In contrast to traditional PCR, where the presence or absence of a DNA sequence is analyzed upon reaction completion, quantitative PCR, or qPCR, monitors DNA amplification as the reaction progresses. In qPCR assays, reaction progression is monitored by an increase in fluorescent signal. The fluorophore can be a double-stranded DNA binding dye, most commonly cybergreen 1, or covalently attached to a target-specific probe, as in TaqMan assays. In cyber assays, the dye fluoresces when bound to double-stranded DNA, but displays weak fluorescence in the presence of single-stranded DNA. This results in an increased fluorescent signal as the reaction progresses, and double-stranded DNA is formed. The most common probe-based assay is a 5' nuclease assay, known as TaqMan. In TaqMan assays, the 5' flapped endonuclease domain of Taq DNA polymerase cleaves a target-specific probe, separating a fluorophore on the 5' end from a quencher on the 3' end, resulting in an amplification-dependent increase in fluorescence. A thermocycler, coupled with a fluorimeter, modulates the reaction temperature during DNA amplification, while also monitoring the fluorescent signal at each PCR cycle, generating a real-time qPCR curve. There are 4 distinct phases within the amplification curve: lag, exponential, linear, and plateau. During the lag phase, amplification occurs, but the signal is not strong enough to be detected above background. The exponential phase is used for quantification because it produces a fluorescent signal measurable over background and contains an abundance of reaction components, permitting a detectable doubling event at every PCR cycle. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle, or CQ value, can be determined. CQ values can be used to evaluate relative target abundance between 2 or more samples, or to calculate absolute target quantities in reference to an appropriate standard curve. qPCR can also be modified to detect and quantify RNA. Interpretation of qPCR data requires careful experimental design and analysis. For parameters on designing and reporting qPCR experiments, please read the MICE guidelines, which have been established to ensure consistent experimental design and data analysis appropriate for peer-reviewed publication.